Hello, everybody. Welcome to Fiveable Crams. We are live. Hi. See people starting to say hi in the chat. Hello, welcome. My name is Jenna. I am your teacher host. We've got a really fun AP Cram preview session for you today for Fiveable Crams. We're going to get started and walk us through.
Awesome. Hi, everybody. My name is Sean, and I am your senior student host. I'm super excited to be here with you guys today. I'm a sophomore at Harvard, and I study psychology. And we're going to get you guys prepared and ready for this exam. Before we get into the content, though, we just have a couple community agreements that we like to have. First, make sure we're all supportive and lift each other up. Let's all be nice to each other, kind, supportive in the chat. Be patient. We'll have 5 nights together during the regular streams, and we have 2 of these preview nights together. So we're gonna get through as much content as we can. You just have to trust us and let us do our thing. We promise you we'll get you through as much as we can. And then be kind. We're all real people. I know I'm a real person. I'm pretty sure from what I know from my fellow hosts, you are all real people as well. No robots on the call. So let's all just be kind to each other. All right. Then we got a couple notes on how we can make the most out of a cram. A couple great ways to just to take away as much as you can from the cram. Take notes while you can, but remember, notes are reminders. Notes aren't write down everything on the slide because actually we have the slides right here for you. So you can check out our slides right down there if you need to see the slides at all with that blue button. So take notes on like maybe tips that the hosts add in. I know AP Psych's all about specific examples, so snag those when you can. The My Stuff tab has all the slides, review packets, and replays for you, so you will have access to all the materials you see tonight. The group chat is a great way to connect with your fellow students. That way you can share tricks, you can share tips, and you guys can help each other out while getting ready for the exam. We're one big AP Psych community for this. Then we also, I just wanted to point your attention to our amazing Q&A tab. Our Q&A tab is where you can drop any and every question you have about AP Psychology, and we will answer as many of those as we can for you. So without further ado, let's introduce you to our hosts that are going to be presenting tonight. First, we have the wonderful Jessica.
Hey guys, my name is Jessica Bakar. I'm a first-year student studying psychology at McGill University, and I'm super excited to be one of your hosts for this preview tonight, as well as the 5 nights for cram mode. I can't wait to embark on this journey together, and you all are going to crush your exams.
Ooh, awesome. Now we have the amazing Victoria up next.
Hi everyone, my name is Victoria Campbell. I'm a first-year student at the University of Alabama. Roll Tide. I'm so excited to be here with you guys tonight. This is the best AP students, and this is the best subject ever, so good luck.
Awesome. And last but certainly not least, we have the fantastic Sushant.
Hey guys, my name is Sushant. I am a senior in the Alliance Academy. I am going to Georgia Tech this fall, so I am not yet a college student, but all my other coworkers are amazing, and I'm very excited to be with you guys here today, and you guys are all going to get a 5 this year.
Awesome. And last but not least, you met her at the beginning, but we have the amazing Jenna Lunge, who's back with us this year.
Yeah, hi, I am Jenna. I'm a PhD candidate at Oakland University, which is just outside of Detroit, Michigan. You probably heard of us recently if you are a college basketball fan. No, we are not in California. We are outside of Detroit. I am defending my dissertation in the fall of 2024, so very close to being a PhD student or a PhD owner, I guess. And this is my second year being the teacher host for AP Fiveable Cram, so with AP Psychology. So I am really excited to be back with you guys. And yeah, let's get it started.
I actually only very recently discovered, after knowing Jenna for like a year and a half now, that Oakland University is not in California.
I know.
But yeah, and then also before we get into it, I want to shout out our fantastic slide designers who created all the materials you're going to see, Mindy Ding and Kate Locke, who aren't with us tonight, but they created all the stuff that you're seeing on the slides.
Thank you, Mindy and Kate. We love you.
All right, before we get into it, follow Think Fiveable on socials. We have LinkedIn, we have YouTube, we have TikTok, we have Instagram to stay in touch with us and to follow along during this cram season. Without further ado, we're about to get into the into the content, but make sure you guys choose to unlock Cram mode. Cram mode is the fantastic resource that Fiveable has to get ready for your AP exams and the SAT and ACT for those of you who need it. I know a bunch of schools are changing back to having those no longer be optional. So you can unlock Cram mode for $10 a month or $40 a year. It'll also give you access to our 5 nights of streams. And yeah, I'll pop a link on the screen for that in just a second. Without further ado, let's get into our Unit 2 preview.
Yay! So I'll be starting off this Unit 2 preview, and today we'll be covering a range of topics from the nervous system to the neuron, the action potential, and neurotransmitters. But before we get into the nitty-gritty of the anatomy of a neuron, different types of neurotransmitters, it's really important that we understand the map of the nervous system. And I would like to walk you through that map today so we know where we're going in future slides. To start off simple, we can break the nervous system down into 2 parts. As you'll see on the screen, this is the central nervous system and the peripheral nervous system. Drop it in the chat right now if you already know what the central nervous system does. I'll give you a few seconds to do that. But if you don't know already, the central nervous system basically encompasses 2 things, the brain and the spinal cord. They are very, very important to the functioning not only of your body, but to consciousness and controlling how we interact with the environment. The central nervous system only does encompass those 2 things, but they really are the mastermind behind the entire nervous system. Now, there are actually 2 types of neurons located only within the central nervous system that I want to make sure that you know today, one being the interneuron that has its soma and axon located only in the central nervous system. These are important to facilitate communication between sensory neurons and motor neurons, as well as projection neurons, which are also located only within the central nervous system. They have their somas located within the CNS, like I said, and they also project their axons, like the name says, projection neurons, to different regions of the central nervous system. On the other hand, everything else that I'm about to talk about falls under the peripheral nervous system. The peripheral nervous system can then be broken down into 2 more parts. And the peripheral nervous system is everything else that isn't the brain and spinal cord. It encompasses your 5 senses that allow us to interact with the world, but it also encompasses a lot of internal functions and internal regulations that we don't think about in our day-to-day lives. So the first distinction that I want to draw within the peripheral nervous system is the somatic peripheral nervous system versus the autonomic peripheral system. In terms of the somatic peripheral system, you may have been taught that this encompasses voluntary actions as well as the system that allows us to interact with our external environment. And that is true. The somatic peripheral nervous system encompasses all 5 of our traditional senses that allow us to take a look at our environment, take in that information, and it sends it to the central nervous system to then allow us to decide how we should interact with our environment. I'll give you one example of something that would fall under the jurisdiction of the somatic nervous system. So let's say that you are walking outside and it's a beautiful day, and then you happen to see this red thing out of the corner of your eye, and you turn and it looks like it's an apple. So the very beginning of that perceptual process would fall under the jurisdiction of the somatic peripheral nervous system. You are using the sense of vision to voluntarily, first you see what is out there in your environment, and then you voluntarily turn your head to get more information about what is out there in your environment. So therefore, you can see that the somatic system has to do with voluntary action as well as interactions between your body and the external environment. On the other hand, the autonomic nervous system takes over functions that you don't really have to think about. You may have been taught that autonomic processes are those that are involuntary, as well as those that have to do with the regulation of your internal environment. And those things are true. Unlike the somatic nervous system that has to do with voluntary functions. You don't have to think about digestion, the regulation of breathing, of heart rate. All of those things would fall under the autonomic branch. A really good way to remember this is autonomic sounds like automatic, and that's exactly how it is. Finally, we can break the autonomic nervous system down into two more parts. There are a lot of parts here, guys, but hopefully this map makes it really easy for you to visualize how we can break down the nervous system. We can break the autonomic nervous system down into two parts, being the sympathetic and the parasympathetic nervous system. The sympathetic nervous system, yes, I see it in the chat. Sympathetic is for fight or flight. That makes me so happy that you guys already know this. You guys are going to crush the exam. Yeah. And as Victoria has said in the chat, sympathetic, you can think of stress. It is when your body is reallocating energy reserves to keeping you safe. The sympathetic nervous system is activated when you are in danger or you are potentially in danger, and it is directing energy from inessential functions to the basic survival functions. You will probably feel that your pupils will dilate, your palms might get sweaty, you might start breathing heavier, your heart rate might increase, all to mobilize the body to keep you safe in times of emergency or danger. Does that make sense? So the parasympathetic nervous system, on the other hand, as Victoria stated in the chat, you can think of parasympathetic taking over in times of peace. A good example of when your parasympathetic nervous system would be dominant would be when you're just chilling in bed, maybe watching Netflix. There's no danger. Your survival's going great. You're not in a state of fight or flight. In fact, you're in a state of rest and digest, or feed and breed. The parasympathetic nervous system is really taking over when you're in a normal state of affairs, and it really is allocating energy resources to quote-unquote inessential functions like digestion, like salivation, like urination, defecation, sexual arousal, all of these things that are important to your longevity. But if you were to be placed in emergency right now, your body would say, no more. We can't be thinking about how we're thirsty. We can't be thinking about how we kind of need to go to the bathroom right now. We need to escape a bear that is chasing after us. So there, I hope you can understand the difference between the sympathetic and parasympathetic. One is for stressful situations, one is for peaceful situations. And overall, that is the map of the nervous system. And we are ready to understand the rest of it and all of the different cells that make it up.
All right, awesome. Thank you, Jessica, for that very well explanation about our nervous system. All right, everyone, now we're going to talk about our neurons, or nerve cells. As we all may know, our neurons are the basic functional units of the nervous system, and we use them in order to send nerve impulses and chemical signals back and forth from the brain through the spinal cord and to the nerves throughout the rest of the body. I like to think of them as kind of our communication networks made up of over 1 billion neurons. So let's break down the neuron. First, we have our dendrites. I like to call them tentacles, but you can call them whatever you like. And they are essentially responsible for receiving signals from neighboring and surrounding neurons through receptor sites. Next, we have our soma. I like to prefer to call it the cell body, as it contains our nucleus, which holds up the genetic information and material for the cell to develop and for the function of the neuron. Essentially, next we have is our axon, and our axon carries electrical impulses away from the soma and to the rest of the neuron through the terminal branches. There are the small kind of dark red circles, and this essentially helps the communication within the brain and for the rest of the body. Think of the axon as sort of a large cable that allows the cell to send information away from itself and to the other cells. And the information being sent down to the axon is in the form of an electrical impulse. And this is a process called action potential, which we'll talk a little bit more about later. Now, protecting the axon is the myelin sheath, which type down in the chat if you know what the myelin sheath is composed of, what type of cells make it up. Essentially, the myelin sheath insulates the axon to help protect the neuron cell, and it speeds up the transmission of electrical impulses down the axon. I think I saw the answer. It is glial cells. So good job with you guys. Now, the gaps in between the myelin sheath are called the nodes of Ranvier. Now, this is where the axon is exposed, and it also helps to promote and continue axon potential, which is a term we're going to talk a little bit more deeply about later. All right, so continuing down the electrical impulses, we will reach the axon terminal, approaching the synapse. The synapse is the gap or the meeting point between two neighboring neurons. In this stage, the action potential releases neurotransmitters into the synaptic gap for the neighboring neurons to pick up, the neighboring neurons' dendrites to pick up. Quick fact, something to keep in mind, is that our neurotransmitters are located at the end of the axon terminal. And the axon terminal converts the electrical signals into a chemical one, and the neurotransmitters enter the synaptic gap.
All right, now that we have the anatomy of a neuron under our belt, we can start talking about how neurons send signals within a neuron and between neurons. So, like Victoria already laid out for us, communication within a neuron is an electrical phenomenon, and communication between neurons is a chemical phenomenon done through neurotransmitters. And to do this communication between and within neurons, we need something called an action potential. But before we can even begin to talk about an action potential, we need to set up what resting potential is. A resting potential is when a neuron essentially is at rest. It is not sending a signal down its axon. It's not releasing neurotransmitters. Now, the resting potential of a neuron rests at between -70 to -90 millivolts. The exact biology of this, why it is in this range, you don't exactly need to know, but the prototypical neuron is about -70 millivolts. And how we get to this negative charge is the work of two factors. First, there's the sodium-potassium pump that concentrates sodium 15 times more on the outside of a neuron and 30 times more for potassium on the inside of a neuron. Now, this sets up a concentration gradient for the prototypical neuron that does not change at all, whether we are experiencing action potential, whether we are experiencing depolarization, hyperpolarization, whatever it is, unless a cell dies. There is always going to be 15 times more sodium outside of the cell and 30 times more potassium inside of a neuron. And you don't actually need to know the 15 versus the 30. The two things that I want you to take away from this are the cations that are necessary to set up an action potential are potassium and sodium. Remember those. Those are good things to remember, guys. So we've set up our concentration gradient. Now, how the heck do we get to a charge of -70 millivolts inside of a neuron? Well, I have one word for you, or I guess maybe three words: potassium leak channels. We have a bunch of potassium all concentrated within a neuron, right? 30 times more inside than outside. But because we have these open potassium leak channels that essentially have an open door policy, it lets potassium go out as much as it wants. And just by the mere force of diffusion, potassium that is in an area of high concentration inside of the neuron is going to want to leave and go outside of the cell to an area of low concentration. And because we have many cations leaving, that is going to leave the cell resting at a charge of about -70 millivolts. So that is our resting potential, and this is the charge that a neuron is going to stay at before it is acted upon by an action potential that I will let Victoria explain beautifully.
Yeah, so as we kind of talked about it a little before with action potential, essentially it occurs when a neuron sends electrical impulses down an axon and away from the cell body or the soma to the remaining parts of the neuron. A quick question for you guys, I want you guys to answer in the chat. What is the ion that depolarizes the potential? Let's see if you guys know that one. But For a neuron to transfer from resting potential to action potential, the process of depolarization must occur. For a neuron to depolarize, there has to be an outside stimulus. Now, this stimulus will create I like to say, domino effect of more positive ions crossing the cell membrane and entering the neuron, which hopefully will trigger the potential for the action potential. However, it must meet that threshold to trigger the action potential. And if not, then there's no action potential that will occur and the neuron will return back to a resting state. This kind of reminds us that neurons act in an all or nothing type of phenomenon behavior. Now, let's say we do have action potential and we go through that process. How do we get back to resting potential? This is through the process called repolarization. And repolarization moves the positive ions back across the cell membrane and out of the neuron in order to rebalance the cells. Now let's talk about the refractory period, which is that short amount of time when no other action potentials can occur until the axon is back to its resting state. Remember that only one reaction can happen at a time. Therefore, the refractory period is that small short of window when we're finishing up our action potential and we're trying to get the neuron back to its rebalancing of a resting state. All right, so now we are going to move over to neurotransmitters. And as Shazam gets started with that, please remind me in the chat where I said the part of where you can find neurotransmitters in what part of the neuron.
All right, that was perfect, Victoria. Great job. Victoria said just 2 slides ago, if you saw, that these neurotransmitters are found in the axon terminal. Axon terminal is the very, very, yes, perfect, Caitlin, is the very end of your neuron, right? And the neurotransmitters that are found there attach to receptors on the dendrite of the next neuron, because neurons are kind of like electrical lines. Kind of think of them. Signals go from one neuron to the next neuron to the next neuron to the next neuron. So one neuron has an axon terminal that excretes neurotransmitters that attach to receptors on the dendrites. Now, these neurotransmitters kind of act like a communication channel for these neurons. So what happens is that the brain, if it wants to create a sense of relaxation, it might increase the amount of GABA, which is a neurotransmitter that creates relaxation in the mind. If it wants to create action, it might, or it wants to create a good mood, it might increase the production of serotonin, and those receptors are going to attach to the dendrites on the neuron. So there are a couple neurotransmitters that are affected by psychoactive drugs. Think like heroin or like cocaine or like these bad type of drugs, like illicit substances, or just coffee or like alcohol, things like that. Those affect neurotransmitters. So there's two types of those. There's agonists, which increase the action of neurotransmitters. So SSRIs, which are a type of antidepressant, increase your serotonin. And serotonin is important in mood. So when you take SSRIs, you don't feel unhappy. So that's what helps. That's what SSRIs help you do. Antagonists decrease the action of a neurotransmitter. So, for example, Botox will block acetylcholine receptors, which acetylcholine is important for muscle contraction. So those muscles don't contract anymore, and Botox basically prevents wrinkles, right? So that's how the antagonists work. A really easy way to remember these two is antagonists are in literature are the villains of a story, right? So antagonists prevent the hero from doing what they want to do. So let's say the neurotransmitter is that hero, it wants to increase some reaction, that antagonist is going to prevent that action, whereas agonists are just the opposite of antagonists. So that's a really easy way to remember those two.
Yeah, awesome. Thank you. I'm loving your guys' favorite neurotransmitters in the chat. Raya, that was a funny one with your glutamate. Okay, so now we're going to talk about the chemical messengers within our body, otherwise known as neurotransmitters. Our first one is our acetylcholine, which Fun fact, well, my favorite fun fact, was the first neurotransmitter discovered. It is also the main neurotransmitter in the brain, as it plays a role in improving attention, memory, learning, and it also triggers muscle contractions. It can also control the heartbeat and stimulate saliva and sweat production. Low levels of acetylcholine can create issues relating to memory and thinking, for example, Alzheimer's disease. Next, we have dopamine, which is my favorite neurotransmitter, as it's important for memory, learning, behavior, regulating blood flow. This neurotransmitter is also known as a pleasure and reward neurotransmitter, as the brain releases dopamine during pleasure activities. Dopamine is also one of the transmitters that is unknown to be a catecholamine. Next, we have moving on is our GABA neurotransmitter, otherwise known as the GABA aminobutyric acid transmitter. And this is the most common inhibitory neurotransmitter in the brain, as it is a mood regulator. So low levels of it can be contributed with anxiety, depression, and schizophrenia. Next, we have glutamate, which is Raya's favorite neurotransmitter, and it plays a major role in learning and memory, and it's important for the brain to function. Next, we have our norepinephrine neurotransmitter, which is released, also sometimes released by some autonomic nervous systems in the peripheral nervous system. Lastly, we have serotonin, and serotonin helps regulate mood, behavior, sleep, and memory, and low levels of this can contribute to common things such as seasonal depression.
All right, so now we're going to go on to drugs and their effect on the brain. So this is something that a lot of people know, a lot of people don't know, but what happens is the first thing that happens when you take a drug is that your tolerance for that drug increases. So let's say you take or somebody takes alcohol. The first time is that someone drinks, they don't really feel they get, it affects them immediately. It's a really high response. And then the next time they take it, maybe they have to take 2 drinks to get that same effect, or then 5 drinks, then 10 drinks. It doesn't, it keeps on, the tolerance gets bigger and bigger and bigger. And what's happening is that there's receptor changes. The brain adapts. It knows it's going to get that alcohol. It knows it's going to have that response. So it diminishes that response to the drug by decreasing the amount of receptor sites. So the same dose has less of an effect. And now you need a higher dose. You need to take more drinks. You need to take, if it's coffee you're drinking, maybe you have to take more coffee to get that same effect. When you're having those late night study sessions or late night, I don't know, gaming sessions, you're taking more coffee to actually be awake. The next thing that happens is, well, dependence. As your tolerance increases on a substance, your reliance on that substance for normal function also increases. So this is a chemical dependence. This is not somebody being, I'm taking, I have to take this because I have to take, I'm taking this for fun. Now at this point, it's becoming a dependence. You're going to have problems if you withdraw from the substance. So you have aches, headaches, fever, chills, all these things can happen when you are dependent on a substance. And now I'm going to hand it over to Victoria for the next 2 parts.
Yeah, so as Shazanth was kind of saying, dependence is when we start to rely on the substances for normal function, and this can lead to problems of addiction, which can cause problems with focus, our memory, learning, our decision making, and also our judgment. People can experience things such as body aches and fever or chills, but it's important to remember that addiction is a chronic psychological disorder. You see, the drugs can cause a host of changes to parts of our brain that controls our body functions. And these alterations provides the opportunity to have dependence and become addicted to these types of drugs. Now, when the drug supply stops, that is when we enter withdrawal. However, don't fret, the brain can adjust to the absence of the absence of drugs over a course of period of time.
Yeah, to just piggyback off that idea of withdrawal, one way that one professor taught it was interesting in that he framed withdrawal effects as being the opposite effects of what the drug would be. So let's say that you're taking maybe an illicit substance and it makes you feel high and very good, and it gets the endorphins going. Maybe you're taking an opioid or something. If the withdrawal symptoms of that would be the opposite, you wouldn't feel euphoric, you would feel the opposite, like the symptoms that Victoria just said. So that might be another way that you could memorize it like that. And just another fun fact, we talked about tolerance. This doesn't happen in humans, but in mice, and at least in rodents, the opposite phenomenon can happen, and that's called sensitization, where unlike tolerance, you need to take more and more of the substance to get the same effect. Sensitization, it sort of is what the word sounds like. You're becoming more sensitive to that substance over time, and you actually need to take less of it to get that same initial effect, which is just something that's really interesting. So now that we've talked about the nitty-gritty, the chemical and electrical communication that occurs in our brain, we're going to take a little bit of a step back and talk about the brain as a whole. This first part that I'm going to talk about is all encompassing of the cerebral cortex. Drop it in the chat right now if you know what the cerebral cortex is. Is this where conscious awareness takes place? Is this a subcortical structure where we are not conscious, where we are not perceiving things? What's up with the cerebral cortex? Yay! Okay, I'm starting to see it. That yes, in the cerebral cortex, we do have conscious awareness. This is the main processing part of our brain, which maybe is a little misleading because the brain is a giant processing machine, cerebral cortex and subcortical structures. But yes, one thing that you want to know about the cerebral cortex is that this is the quote-unquote conscious part of your brain, and this is where things enter conscious awareness. This is where perception happens. Okay, so let's get into the different parts of the cerebral cortex, shall we? So the first part that we have is the frontal lobe, and as the name states, it is at the front of your brain. A few key functions or things that are localized here that you want to know are speaking, including Broca's area. Does anybody know what Broca's area is and or what happens when you have Broca's aphasia? Just drop it in the chat. If you know, if you don't know, it's okay to take a guess. I would love to see it and love to see you applying your neurolinguistics knowledge to this preview session. Yes. Okay, great. So I am seeing in the chat that you guys are mostly right, that Broca's area is responsible for language production. Again, this is located in the frontal lobe, and this means that if we have Broca's aphasia, we are going to have problems with language production. And aphasia, just generally, it's a term for, in simple terms, problem with speech. There are different types. There are many, many different types. We're not going to talk about them all today. But with Broca's aphasia, it means that your comprehension of words is fine. That's not the problem. You are able to hear what other people are saying. You are able to process it. And you are able to process it and up to the point where you want to articulate it, but you can't articulate it. It is the problem of language production. One way that my psych teacher actually taught me the difference between Broca's aphasia and Wernicke's aphasia is saying that Broca's aphasia, this little mnemonic thing, the mouth is broken. It's actually not a problem of the mouth and its muscles. It's a problem of the frontal lobe. But you can think about it like that, where you want to express yourself. You have the understanding to express yourself. Your mouth, though, you cannot produce the speech in a way that is complete and coherent. And actually, for people with Broca's aphasia, because they can understand, it's a really, really frustrating aphasia to have, because you want to express yourself, but people cannot understand you intelligently because you do not have the capacity for language production due to damage in the frontal lobe. So other things that are localized in the frontal lobe include planning and judgment, which are located in the prefrontal cortex. The prefrontal cortex is very, very interesting, and it doesn't just do planning and judgment, but it has a very significant role in these cognitive functions. Fun fact, the reason why the drinking age in the United States of America is 21 years of age is because your brain and your prefrontal cortex specifically does not fully develop into your early mid-20s. And so the reason why this law is in place federally is to protect yourself, because without a fully developed prefrontal cortex, the fear is that your judgment and your decision-making skills will be impaired, among other things, and you may not make the best decisions while drunk, let alone while sober. Finally, movement and the motor cortex are also localized in the frontal lobe. The motor cortex and movement as a whole is really interesting, and we can understand the interactions and the communications between the central nervous system and the peripheral nervous system to run it back to what we were saying in the first slide with my map of the nervous system. So let's go back to the somatic branch of the peripheral nervous system. Let's say that you spot an apple out in your environment somewhere. You're interacting with your environment. You are activating your five senses and your visual sense. You can see that there is an apple out in your environment. Now, that information, those would be called afferent signals coming from the atmosphere or from the environment through the peripheral nervous system to the central nervous system, to your brain, where eventually it is going to go to your primary visual cortex and extrastriate areas. It's going to be processed and perceived as an apple, and now your central nervous system is going to send efferent or efferent signals exiting the brain back through the central nervous and peripheral nervous system to your skeletal muscles to tell you, hey, go ahead and pick up that apple, go ahead and take a bite. It's going to tell you how to interact with your environment, and the motor cortex here is going to play a role in that, in sending the motor signals through the central and peripheral nervous system, these efferent signals telling you how to interact with your environment. And I hope through that example you can understand a little bit of the relationship with the afferents and the efferents, as well as this communication, this talking between your central nervous system and peripheral nervous system, because that's a lot of what consciousness and conscious awareness and sensation and perception and action is, this back and forth between these two branches of the nervous system. All right, I think that we've covered the frontal lobe enough. Time to get on to the temporal lobe. Can anyone tell me, there are a few ideas up on the screen, but when you think of temporal lobe, what do you think of? Let me know in the chat right here. What do you think of when we think of the temporal lobe? Great, I'm seeing tempo, sound, hearing. Yes, this is all great. I was going to tell you guys that one way to think about the role of audition in the temporal lobe is to look at that little prefix there, tempo, and think of sound, but it looks like you already have that. Psych kids and their mnemonics there on it. Love to see it. So yes, auditory processing does happen in the temporal lobe. And this is where the primary auditory cortex is. This is where we perceive pitch, timbre, all of those characteristics related to sound, where we can identify the differences between different sounds and really where it enters our conscious awareness, right? Where we are able to say, hey, that is the sound of a bell, or that is the sound of a dishwasher. Another function that is localized in the temporal lobe is semantic processing. This is occurring in Wernicke's area. We already talked about Broca's aphasia. Type it down in the chat what Wernicke's aphasia is. It's okay if we don't know too. I know there's also a lag with the chat, but yes, I'm seeing it, great. with Wernicke's aphasia, you are fluent in your speech. This is actually called fluent aphasia, but what comes out is quote unquote word salad, right? Because the people who have Wernicke's aphasia, one, they cannot understand when people are talking to them. They understand when people are talking to them, but they can't understand the actual content of it. And they don't produce coherent, content-rich language either. They can talk and talk all they want, but it sort of sounds like nonsense. And finally, besides semantic processing, long-term memory is also somewhat localized in the temporal lobe. The semantic memory is one form of long-term memory. Actually, if you tune in on Thursday for our second Cram preview, we're going to cover all about long-term memory, just a sneak peek. So semantic memory is located in the temporal lobe, so is episodic memory in the medial temporal lobe with the hippocampus. Other forms of long-term memory that aren't necessarily here in the temporal lobe, that would be implicit memory. That's more in the basal ganglia and prefrontal cortex with habit formation. With the parietal lobe, we have sensory processing in the somatosensory cortex, as well as spatial awareness. One way that I am able to remember that parietal lobe is associated with touch and the somatosensory cortex. That's what somatosensory does. It's a lot of touch in the cutaneous sense. I think about a child and how a parent, like parietal parent, would always tell a child, hey, don't touch that, because kids are really curious and they want to touch everything. So that's a little mental trick for you guys to remember. Somatosensory cortex is in the parietal lobe. And spatial awareness is also in part localized here. And we can see this with the two streams, the ventral and dorsal stream of visual processing, that the dorsal stream is the where pathway when we are analyzing visual stimuli. The dorsal pathway is going to extend from the occipital to the parietal lobe, and in the parietal lobe, this is where things like spatial information and movement are really going to be processed and analyzed. Finally, as we wrap up the cerebral cortex, we have the occipital lobe. You can remember occipital, sort of like the prefix, the OCC, being present in the word ocular, which is related to sight. And here, as you can hopefully tell by the name, we have visual processing going on. And that's really the only thing you need to know about the occipital lobe here. So that is a brief overview of the cerebral cortex and the neuroanatomy of it. Now we're going to get into some subcortical structures and some other structures right now.
All right, let's go into more stuff in the brain. So we're going to talk about two different systems that control memory and also that control automatic function. So the first is the limbic system. And the limbic system controls memory, learning, motivation. It controls a lot of different things that you don't really think about. So the amygdala is your first part of the limbic system, and it controls fear and aggression and your emotions. And it is mostly related to emotional memory. And this is really important on the AP exam. I had a question last year on the AP exam where it was talking about that it had both the amygdala and the hippocampus, and both of them relate to memories, but it was a very emotional memory. And, or at least that's what the question said. So the amygdala would be more responsive in retrieving that memory. So the amygdala is mainly to do with emotions. It's your emotion center. That's how I'd like to think about it. Yeah. In the chat, that's perfect. Amygdala, Amy is mad. Yeah, that's an emotion that you can feel through the amygdala. The next part of the limbic system is the thalamus, and that's a sensory switchboard. So essentially all your senses go through the thalamus, regardless if you are touching, tasting, hearing, or seeing something. It's all going to go through the thalamus, and then it's going to be processed by the brain, other than the exception of smell, which is directly connected to the brain through the olfactory bulb. But all the other senses are connected via the thalamus. The next part is the hippocampus, and the hippocampus is one of the more important ones, right? It deals with episodic memory. It's really important in retrieving memories. And episodic memory is memories that are, well, kind of, well, and in a sense, episodes, right? That's what you think of when you hear episodic memory. So let's say you had a really nice Christmas one year with your parents, and you retrieve that memory, that's an episodic memory. Another part is the hypothalamus, and this is the 4 that you need to know for the AP exam. And this has to do with internal regulation. So there's it mainly has to do with hormones. So there's a lot of different hormones. I'm sure you guys know a bunch of them. So put in the chat what hormones do you guys know about. Chances are, whatever hormones that you guys know about, like testosterone, androgen, all these things, there's a ton of growth hormones. Melatonin is a great as one. Cortisol is one. But all these hormones are controlled by the hypothalamus, and it controls all those different. Yeah, it can. That's perfect. Okay, then we're going to go on to the brainstem. So this controls your automatic functions. So first, we're going to go with the medulla, and the medulla does mostly internal regulation, and it helps you. It also helps with the vomiting reflex. So if ever you're on an airplane and you have to vomit into that nice little plastic bag, paper bag they give you, that is going to be your medulla acting. The next thing is your reticular formation, and it helps direct stimuli, and it's motivates you and it makes sure that you're aroused and actually active. If you don't have your reticular formation, or if it's not working, then you won't be able to function because you won't be aroused. You'd probably be just asleep. You'd probably be in a coma. Another thing you have is the cerebellum, which controls voluntary movement, and it also controls procedural memory. So procedural memory is something that a procedure that you think of, and that's for example, when you drive a car, the driving of the car, you don't really think about driving a car. You just do it. When you turn left, you don't think about, oh, I'm turning left. You just do it. You turn the wheel left and you go. Another type of procedural memory is if you're playing an instrument. You don't really at a point when you play an instrument, you don't really look at the keys anymore. You don't look at the notes you're playing. You just know what notes to play based on based on the procedure. So that's how you know that. And the cerebellum is often called the little brain. It also does a lot of other important things like balance. So if you're on a tightrope and you want to balance, it makes sure that you're able to balance on that tightrope, and it controls that. And the last one, not least one, is the pons, and it helps in your coordination and mainly your sleep. So make sure you're able to sleep. It also controls your breathing, things that you don't think about. Basically, anything, the medulla and the pons basically control things that you do not think about on a day-to-day basis. Now that I mentioned breathing, you guys are probably all not automatically breathing anymore. You guys are probably all breathing normally through your like conscious mind, but eventually you will breathe normally again, right? You'll breathe automatically. So those are the things that the medulla and pons will control, and the rest of the brainstem also controls automatic things. If you ever think of the brainstem. All right.
Just to add on to that a little bit, I have a fun little mnemonic, I guess, to remember the hippocampus. It's a pretty important part of the brain, but if you're having trouble remembering what it does, my AP Psych teacher told me, think of a hippo on campus, like a giant purple beast just walking around on your high school campus. That would be a crazy event to remember, right? And so that hopefully can help you remember that episodic memory is for vivid, event-related, personal, autobiographical memories. All right, so now we have some practice MCQs for you. I think that we have 2 lined up. So the first one, damage to Broca's area is most likely to impact an individual's ability to A, encode episodic memories, B, encode semantic memories, C, produce and articulate language, D, understand and comprehend language, or E, reason critically in a stressful environment. Great. So as I am seeing from the responses in the chat, you guys have absolutely crushed this question. And as we can see on the screen, and as I talked about a little bit with the neuroanatomy stuff, Broca's area is important for the production and articulation of language. Therefore, damage to this area, as in the case of Broca's aphasia, would lead to an inability and reduced ability to produce and articulate language. And yes, D would be Wernicke's aphasia or Wernicke's area. Cool. So for our second practice question, following a neuron's firing, it enters a blank that aims to blink. Okay, so I see that we are getting some answers in the chat. We do also have the poll function, and if you guys could vote in there, that would be great, just so we can see the breakdown. It's a little bit easier for us. So I will wait a little bit more before revealing the answer. Keep rolling those votes in through the polls if you can. All right, let's go ahead and reveal the correct answer here. And as you can see, the correct answer is B. I think the second most voted answers were A and D. I can go over that quickly. So the correct answer is B. After experiencing an action potential after a neuron fires, it enters a refractory period. This is the official name, the vocab term that you want to jot down, that you want to remember. You can kind of think of it as a recovery period, but it's not actually called that. It's called a refractory period. And during this period of about 1 millisecond, a neuron that is just fired an action potential cannot fire another one. It needs to, and as the second part of the question states, reestablish its resting potential. So I understand why you might have chosen A. It's just really a semantic issue. The proper term for this repolarization is called a refractory period. And D was also another frequently chosen answer. In reestablishing a resting potential, we're not necessarily depolarizing the neuron. Actually, what happens during a refractory period, the neuron becomes hyperpolarized. So we're not actually fully depolarizing it to -40 that we would experience in an action potential. We are just letting the processes of diffusion occur so that we get back down to our resting potential of about negative millivolts, if that makes sense. And that's why the correct answer is B. All right, so that is it for the content today. It looks like we have a practice quiz lined up for you guys.
Yeah, wonderful. Thank you guys so much for all that wonderful information. We're going to go ahead and I believe have Shant come give us the practice quiz. So I will stop sharing my screen. So how are we feeling? Do we feel like we learned a lot about neuroanatomy? How are we feeling in the chat, guys? I saw some questions about like what's this like in relation to the actual AP exam. Those questions are two very similar ones. All of our questions are adapted from questions. Oh, sorry, my lights are automatic in here. Are either adapted from the AP exam or designed by myself and the wonderful Jenna, who and Jenna teaches Psych 1, which is very similar to AP Psychology. So these are all similar to questions you are going to see. All right, let's get this quiz rolling. All right, I see 40 in there. You guys should be able to continue to join as we start it. So let's go ahead and start that quiz.
I'm going to interrupt real fast. Can we have somebody post the link to the quiz because the QR code is about to disappear, please?
All right, so for our first question, it looks like a good proportion of you guys have already answered. So I think we can reveal the answer if we want to do that. The question is, in psychological research, what would be the most appropriate method to gather detailed information about an individual's life, history, and personal experience? Wow, 81% of you got this right. The correct answer is a case study because we are looking at one individual. Yes, the key is one individual. Yes. All right. Next question. Yes. Okay. I love the samba music. Great to hear it. Okay. Which perspective in psychology would most likely explore how our interpretation of a situation affects how we react to it? All right. I think we can go ahead and reveal the answer now. Great. Again, you guys crushed this question. So in looking at how someone interprets a situation, we're going to be looking at the cognitive perspective, which has to do with mental processes. And in terms of this, it would be our expectations and top-down processing that we bring to a situation. All right, next question. We're going to do maybe two more since we're nearing our hour. And even though these questions didn't fully review what we went over today, it's still a really great review for you guys. And when you're taking that exam, it might feel like whiplash a little bit. You're going to be going back and forth between these MCQs, pulling from different content areas. So this is good practice for that. And make sure to unlock Cram mode and come back to see us on nights 1 through 5 to get all of this content reviewed and gone over, polished up for that big day. Yes, the music is fire. All right, I think we can reveal the answer. Great. This question was a little bit longer. It pulls on some a bit of statistics knowledge, but it's great to see that the majority of you got this one right. And I think that we will be wrapping up our hour right around now. Thank you all for joining us so much.
Thank you all so much for coming to our first Cram preview. We are really excited to have had you. We have one more Cram preview that you can sign up for before we actually have our regular Cram nights. So you can follow Think Fiveable. Sean, if you have any other plugs you want to throw out there right now, would be awesome. But yeah, we appreciate you guys so much. And we're really looking forward to cramming with you guys.
Yeah, thank you folks so much for joining us tonight. Like we talked about earlier, make sure you unlock that cram mode. We'll have tons of practice questions, tons of FRQs, again, written by someone who teaches it all. Amazing Jenna over here. So you know that you're gonna get a lot of knowledge out of that. Also, it would be super helpful to us if you could fill out this quick survey down at the bottom. It's linked in that green button below. If you can just take a minute or two, let us know what you want more of, what went well tonight, what maybe didn't go so well, and how we can best prepare for the next preview and for the week of crams that we hope to see you all at. But
Last thing is if a little preview, if you sign up for Cram modes, I do a psychology show and tell every night, and I'm going to give you a sneak peek at next week's or whenever it is. This is my not show and tell for psych stuff, but my friend's mug, which gets me through PhD. So when it comes to the actual crams, we will have psych related PhD show and tell.
also, yeah, it's one of my favorite parts of every cram season. There's some good ones. I promise you guys. I promise you guys. So thank you again so much for joining us. Remember, $10 for the month for cram mode or $40 for the whole year unlocks everything in Fiveable's library, thousands of practice questions. Plus, you get to spend 5 more nights with us, which I think would be a lot of fun. So with that, have an awesome night, folks. Please fill out the survey, and we will see you all on Thursday. Bye, everybody.